Hello friend, welcome to my YouTube channel, Gajpal A Learnings. In this video we are going to continue the previous topic, that is Introduction to Cellular Level of the Organization, which is a part of B Pharmacy first semester, Human Anatomy and Physiology Part 1. Then to be covered in this videos are cell junction, general principle of cell division, intracellular signaling, intracellular signaling pathway activation by extracellular signal molecule, and forms of intracellular signaling. So let's start. First of all, let us know what the cell junction means. Cell junctions are specialized structures that facilitate cell-cell adhesion, communication, and tissue organization in multicellular organisms. They are found in various tissues and serve different functions. Here are three major types of cell junctions. First, tight junctions. Tight junctions, also known as occluding junctions, are located at the apical top portion of epithelial cells. These junctions form a barrier that prevents the passage of molecules between cells, effectively sealing the spaces between cells. Tight junctions accomplish this by tightly connecting adjacent cell membranes through the interaction of transmembrane proteins, such as claudins and occludins. The main function of tight junctions is to regulate the movement of substances through the paracellular pathway, ensuring selective transport across epithelial tissues and maintaining tissue integrity. Second, adherence junctions. Adherence junctions are found below tight junctions and provide mechanical support and adhesion between adjacent cells. They consist of transmembrane proteins called cadherins that link to the actin cytoskeleton inside the cells through a complex of intracellular proteins, including catenins. Adherence junctions promote cell-cell adhesion, reinforce tissue structure, and transmit mechanical forces between cells. They also play a role in cell signaling and tissue development. Third, gap junctions. Gap junctions are specialized channels that allow direct cell-to-cell communication and the exchange of small molecules, ions, and electrical signals. They are composed of proteins called connexins, which form channels called connexins. Connexins on adjacent cells align to create a pore-like structure that permits the passage of substances. Gap junctions are crucial for coordinating cell activities, such as the synchronization of electrical impulses in cardiac muscle cells and the spread of signals in the nervous system. They are particularly abundant in tissues requiring rapid communication, like the heart and the nervous system. Fourth, desmosomes, or spot junctions. Desmosomes are cell junctions that provide strong adhesion and mechanical strength between cells. They are commonly found in tissues that experience mechanical stress, such as the skin and cardiac muscle. Desmosomes consist of transmembrane proteins called desmogleins and desmocollins, which connect adjacent cells, and intracellular proteins called desmoplakins, which anchor the cytoskeleton to the cell membrane. Desmosomes create spot-like attachments, forming a network of strong connections that distribute mechanical stress across tissues. Fifth, hemidesmosomes. Hemidesmosomes are specialized junctions that anchor epithelial cells to the underlying basement membrane, a thin extracellular matrix layer. Hemidesmosomes are primarily found in tissues subjected to mechanical stress, like the skin and the lining of blood vessels. They contain integrins, transmembrane proteins that connect the extracellular matrix components. Example: laminin in the basement membrane to intermediate filaments, such as keratin, within the cell. Hemidesmosomes provide stability and anchorage to epithelial tissues, helping to resist forces and maintain tissue integrity. Sixth, focal adhesions. Focal adhesions are dynamic cell junctions that connect cells to the extracellular matrix. They are present in many cell types and play a crucial role in cell migration, tissue development, and wound healing. Focal adhesions involve integrin transmembrane proteins that bind to specific extracellular matrix proteins such as fibronectin. Inside the cell, focal adhesions connect to the actin cytoskeleton through a complex of proteins including talin and vinculin. Focal adhesions serve as points of attachment, transmitting mechanical forces between the cell and the extracellular matrix, and are involved in signaling pathways that regulate cell behavior. In short, we can say that cell junctions are essential for maintaining the structural integrity of tissues, allowing cells to function cooperatively and coordinate their activities. They contribute to the overall organization and function of multicellular organisms. Next topic is general principle of cell communication. Cell communication, also known as cell signaling, is a fundamental process by which cells communicate with each other to coordinate their activities, respond to environmental cues, and maintain proper tissue function. Here are the general principles of cell communication. First, ligands and receptors. Cell signaling typically involves the interaction between signaling molecules called ligands and specific receptors on the surface of target cells. Ligands can be hormones, growth factors, neurotransmitters, or other signaling molecules. Receptors can be membrane-bound or located within the cell, depending on the nature of the ligand. Second, reception. When a ligand binds to its corresponding receptor, it triggers a specific biochemical response in the target cell. The receptor may undergo conformational changes, leading to the activation of downstream signaling pathways. Third, signal transduction. Signal transduction refers to the process by which the signal from the ligand-receptor interaction is relayed from the cell surface to the cellular interior. This involves a series of molecular events, such as the activation of intracellular signaling molecules, amplification of the signal, and the initiation of specific cellular responses. Fourth, intracellular signaling pathways. Intracellular signaling pathways are complex networks of biochemical reactions that transmit and process the signal initiated by the ligand-receptor interaction. These pathways involve the activation of various proteins, including kinases, phosphatases, second messengers, and transcription factors. The signal is often amplified at multiple steps in the pathway to ensure an appropriate cellular response. Fifth, cellular response. The ultimate outcome of cell signaling is the activation of specific cellular responses. These responses can vary widely, including changes in gene expression, alteration of enzyme activity, cytoskeletal rearrangements, secretion of molecules, cell growth, differentiation, or cell death. Sixth, termination and feedback. Cell signaling processes include mechanisms to terminate or attenuate the signal to prevent overstimulation or prolonged activation. Feedback mechanisms, both positive and negative, regulate the intensity and duration of signaling events, ensuring appropriate cellular responses and maintaining cellular homeostasis. After studying the above content, we can say that cell communication is essential for proper development, tissue maintenance, immune responses, and physiological processes in multicellular organisms. Dysregulation of cell signaling can lead to various diseases, including cancer, autoimmune disorders, and neurological disorders. Understanding the principles of cell communication is crucial for advancing our knowledge of cellular processes and developing therapeutic interventions. Next topic, intracellular signaling. Intracellular signaling, also known as cell signaling, refers to the process by which cells communicate with each other to transmit information and coordinate their activities. It involves the transmission of signals from the extracellular environment to the intracellular compartments of cells, leading to specific cellular responses. Intracellular signaling is crucial for the regulation of various physiological processes, including cell growth, differentiation, metabolism, cell survival, and responses to external stimuli. It allows cells to sense changes in their environment and respond accordingly, ensuring proper functioning and homeostasis within the organism. The process of intracellular signaling typically involves the following steps. First, reception. Cells receive signals from the extracellular environment. These signals can be in the form of chemical molecules, such as hormones, growth factors, neurotransmitters, or other signaling molecules. Second, signal transduction. The received signal is transmitted from the cell surface to the intracellular compartments. This involves a series of molecular events and signaling pathways that relay the signal from the receptor to the appropriate intracellular targets. Third, amplification. In many signaling pathways, the signal is amplified at various steps to ensure a robust and effective cellular response. This amplification step often involves the activation of multiple proteins or enzymes in a cascade-like manner. Integration and modulation. Different signaling pathways can converge or interact with each other, allowing cells to integrate multiple signals and modulate their responses accordingly. This integration and modulation helps cells achieve appropriate and context-specific responses. Fourth, cellular response. The intracellular signaling pathways ultimately lead to specific cellular responses. These responses can vary widely and depend on the nature of the signaling molecule, the cell type, and the specific signaling pathway involved. Cellular responses can include changes in gene expression, alterations in enzyme activity, cytoskeletal rearrangements, cell proliferation, differentiation, migration, or programmed cell death, apoptosis. In short, we can say that intracellular signaling is a highly regulated and complex process that involves a network of interacting molecules, including receptors, kinases, phosphatases, second messengers, transcription factors, and other signaling proteins. Dysregulation of intracellular signaling can lead to various diseases, including cancer, autoimmune disorders, metabolic disorders, and neurodegenerative diseases. Understanding intracellular signaling mechanisms is critical for advancing our knowledge of cellular processes, disease mechanisms, and developing therapeutic strategies that target specific signaling pathways to treat various diseases. Next, intracellular signaling pathway. Activation by extracellular signal molecule. Intracellular signaling pathways are activated in response to extracellular signal molecules, also known as ligands or signaling molecules. These extracellular signals can be diverse, including hormones, growth factors, neurotransmitters, cytokines, and many others. The activation of intracellular signaling pathways typically involves several steps. First, ligand-receptor binding. The extracellular signal molecule binds to its specific receptor on the surface of the target cell. The binding may occur through direct contact between the ligand and the receptor or through intermediaries, such as cell surface receptors or co-receptors. Second, receptor activation. Upon ligand binding, the receptor undergoes conformational changes or other structural modifications that lead to its activation. This activation can involve the dimerization or clustering of receptors, phosphorylation of receptor tyrosine kinases, or other mechanisms depending on the receptor type. Third, signal transduction. The activated receptor initiates intracellular signaling cascades through a process known as signal transduction. This involves the transmission of the signal from the cell surface to the intracellular compartments such as the cytoplasm or nucleus. Fourth, second messengers and amplification. In many signaling pathways, the activated receptor triggers the production or release of second messengers, which are small molecules that help transmit and amplify the signal within the cell. Examples of second messengers include cyclic AMP, CAMP, calcium icons, Ca2-jor, inositol trisphosphate, IP3, and diacylglycerol, DAG. Second messengers can activate downstream signaling molecules and participate in the regulation of cellular responses. Fifth, activation of signaling proteins. The activated receptors or second messengers activate specific intracellular signaling proteins, such as kinases, phosphatases, G proteins, or other adapter proteins. These proteins undergo modifications, such as phosphorylation or binding-unbinding events, which transmit the signal to downstream components of the signaling pathway. Sixth, signal propagation. The activated signaling proteins relay the signal to subsequent components of the signaling pathway, leading to further amplification and propagation of the signal. This often involves a cascade of phosphorylation events or protein-protein interactions, resulting in the activation or modulation of downstream signaling molecules. Seventh, cellular response. The propagated signal ultimately leads to specific cellular responses. These responses can include changes in gene expression, alteration of enzyme activity, cytoskeletal rearrangements, cell proliferation, differentiation, or other physiological responses specific to the signaling pathway and the cellular context. Hence, the activation of intracellular signaling pathways by extracellular signal molecules is a highly regulated process that allows cells to interpret and respond to environmental cues. The complexity and diversity of signaling pathways enables cells to integrate multiple signals, fine-tune responses, and ensure appropriate cellular behaviors and functions. Now the last topic is forms of intracellular signaling. Forms of intracellular signaling are: first, contact-dependent signaling. Contact-dependent signaling occurs when cells physically interact with each other through direct contact. In this form of signaling, the signaling molecule ligand is present on the surface of one cell and binds to its receptor on an adjacent cell. This type of signaling is important during development, immune responses, and cell-cell communication in various tissues. Examples of contact-dependent signaling include the interaction between immune cells and target cells during immune responses, as well as interactions between neurons and glial cells in the nervous system. Second, paracrine signaling. Paracrine signaling involves the release of signaling molecules, ligands, by one cell, which then act on neighboring cells in the local microenvironment. The signaling molecules are secreted into the extracellular space and diffuse over short distances to reach nearby target cells expressing the appropriate receptors. Paracrine signaling allows for local communication and coordination between cells. Examples of paracrine signaling include growth factor signaling during tissue development, immune cell communication, and the release of neurotransmitters in the nervous system. Third, synaptic signaling. Synaptic signaling is a specialized form of paracrine signaling that occurs in the nervous system. It involves the transmission of signals across synapses, which are the junctions between neurons. In this process, neurotransmitters are released from the presynaptic neuron into the synaptic cleft, where they bind to receptors on the postsynaptic neuron. Thus binding leads to the transmission of signals from one neuron to another, allowing for rapid communication and information processing in the nervous system. Fourth, endocrine signaling. Endocrine signaling involves the release of signaling molecules, called hormones, into the bloodstream by endocrine glands. These hormones are then transported throughout the body to target cells expressing specific receptors. Endocrine signaling allows for long-distance communication and coordination of various physiological processes. Hormones can have widespread effects on multiple target cells or specific effects on particular tissues. Examples of endocrine signaling include the actions of insulin, estrogen, and testosterone, which regulate numerous processes in the body, including metabolism, growth, and reproduction. These different forms of intracellular signaling play critical roles in various physiological and developmental processes, allowing cells and tissues to communicate and coordinate their activities for proper functioning of the organism. Thanks for watching this video.